research is something which is a little bit boring.
Okay, the first thing what you said, research can be boring. It is not.
Professor Subhash Chandra Lakhotia is a distinguished Indian cytogeneticist and a lifelong distinguished professor of zoology at Banaras Hindu University.
You're doing it for your own choice. You're not doing it because somebody else wants you to do this thing or that thing. I decided not to work in a research institute, which had its own mandate. I wanted always to be a university teacher, when a teacher, and to do research as I like it.
Why Drosophila plays such a significant role and becomes a model organism and opens altogether a different gateway for the researchers? How it is?
Fly is one of the organs which I would always say that it doesn't need a passport or visa to travel to any country. Firstly, it requires very limited laboratory infrastructure. Food is very simple. Any kitchen can cook it. We don't need anything. Almost 120 years of resources that have been built up and accumulated. See, even the first mutant that Morgan isolated in 1909 is still available. None of the mutants are thrown out. They are maintained and they are catalogued and they are shared. And consequently, the other advantage is that we can make transgenic flies very easily, very conveniently. And the result is now you take any human gene and put it into Drosophila and see where it expresses, what it does, how it does. If we take protein coding genes between Drosophila and humans, 60 to 70% genes are identical. The gene that gives instruction to a cell to become eye is same as in human. Same gene instructs human cells to form an eye, and same gene instructs Drosophila embryo to make an eye with cell. Drosophila eyes are very different from humans.
Yes.
Each Drosophila eye has about 800 lenses. Only one lens.
True.
So the question is, how does it do? And this is where it becomes very convenient to understand what it does to develop. If we compare human and chimpanzee DNA, for example, protein coding genes are almost identical. What differs is non-coding, and that's what regulates. Chimpanzee has a nose, we have a nose, they have 2 eyes, they have face looks similar, but what is the difference? Regulation. Which way the cells will divide, how many times they divide, when it will divide, these are regulatory events. Why each one of us having same 46 chromosomes, same similar genome, look different, it's this non-coding part that makes this. And Drosophila has stuck with me since then.
Okay.